Hi everyone, and welcome to our video about cofactors. So this is the second video in our enzyme series, and what we're going to be doing in this one is having a little look at the spec reference 2.1.4e, which is the need for coenzymes, cofactors and prosthetic groups. First thing then, what on earth do we mean by this word cofactor? When we're talking about a cofactor, we are referring to a substance that has to be present to ensure that an enzyme catalyzed reaction will take place at the appropriate rate. Now, we get two different types of cofactor here. Some are going to be an integral part of the enzyme structure, and those ones we refer to as prosthetic groups. Others are just going to form a temporary association with the enzyme. And these will be things like our mineral ion cofactors or organic coenzymes. So we basically have just those two categories, either permanently part of the enzyme structure or temporary bindings that will only exist for a short period of time. If we go back to some basics on enzymes, hopefully we remember that when we have an enzyme catalyzed reaction, the enzyme and the substrate are going to bind together and form this thing called the enzyme substrate complex. Makes sense? Enzyme and substrate together, enzyme substrate complex. Now, what we actually find is that if we've got certain ions present in the same location as our enzyme and our substrate, it just makes the formation of that ESC more likely. Now, if that's more likely to form, what we're going to find is the rate of reaction will increase. So how can we actually achieve this then? First one is that the cofactors could act as what we call a co-substrate. So what that means is that the substrate itself and our little cofactor are going to combine, and when they're together, they form the complementary shape to the enzyme's active site. So that's one of our options. The next is that the cofactor is going to change the distribution of charges on the actual surface of the substrate or on the surface of the active site of the enzyme. And what we then find is, as a result of that charge change, it makes the temporary bonds between the enzyme and the substrate easier to form. So what we can see here is we've got a couple of different approaches that these cofactors could take to achieve the same end goal. And a good example of one of these cofactors that is using an ion would be amylase. Now, amylase is an enzyme we should be familiar with. It's obviously one we talk about in saliva and the like. But amylase is only going to function if there are chloride ions present. So without chloride ions, amylase won't function. The other option that we talked about at the beginning was that we have these cofactors which are permanently bound to the enzyme. Now, that permanent binding is achieved through the formation of covalent bonds between this cofactor and the enzyme itself, and when that happens, we have what we refer to as a prosthetic group. And an example of a prosthetic group you should know is in the enzyme carbonic anhydrase. Then we will find that that has a zinc ion, Zn2+, permanently bound to the active site. So because that zinc ion is permanently bound to the active site of our carbonic anhydrase, we refer to it as a prosthetic group. In terms of what our carbonic anhydrase actually does, we're going to see this coming up in a future video. But just to give you a little sneak peek, if you will, this is the enzyme that's going to convert the carbon dioxide and water into a substance called carbonic acid. Now, the carbonic acid is then able to break down into protons, which are hydrogen ions, and hydrogen carbonate ions. So what we can see in this little equation here, we're starting off with the carbon dioxide and water. Then we've got a reversible reaction. Remember, this little sign here means it's a reversible reaction, whereby the carbon dioxide and water are going to be converted into the carbonic acid, that's the H2CO3, and then that is going to obviously break down to form our protons and the hydrogen carbonate ions. Now, this whole process is going to be very important when we come to understand how our body is able to transport carbon dioxide. So obviously we know that our respiring cells make carbon dioxide. We know that we're going to exhale from the lungs. We then got to get it from those respiring cells to the lungs themselves. And the way we do that is through this reaction in part. When we talk about these things called coenzymes, then these are just small organic non-protein molecules that will bind to the active site of the enzyme temporarily. Now, this is going to happen usually just before or at the same time that the substrate binds to the active site. What we actually find in this scenario is that the coenzyme itself is going to be chemically changed in this reaction. So what that means is before we can actually use it again, we need to recycle that coenzyme back to the original state. A large number of these coenzymes that we're going to encounter actually are derived from vitamins. So basically you've got to have these vitamins in your diet in order to produce the coenzymes in the correct quantities. Otherwise, you're going to suffer with a range of medical conditions depending on which vitamin you're deficient in. So I've given you just one example at this time of a couple of coenzymes that are going to be very familiar to us before the end of the A-level biology course. We need vitamin B3, so nicotinamide, and that one is going to be used to form these two coenzymes, NAD and NADP. Now we're going to find out why that is important to us in the year 13 content, where we start to have a little look at some of those biochemical processes. If we have a lack of vitamin B3, the nicotinamide in our diet, then what's going to happen is we're not able to form the right amount of those coenzymes, and therefore we're going to suffer with a condition called pellagra. Now, the sort of symptoms that we get there, things like dementia, diarrhea, dermatitis, and you will see why it's quite wide-ranging when you understand a bit more about what the coenzyme is used in. You don't have to memorize all the different vitamins, what they form, and the potential medical conditions that you could suffer if they're lacking. Just be aware that obviously if we do have vitamin deficiency, there's a chance you're not going to be able to make specific coenzymes, and therefore you're going to suffer with certain symptoms. Depending on the vitamin determines the actual symptoms at the end of the day. As always, I suggest that you subscribe to the channel so you can see when I next upload a video, and of course head on over to the A-Level Biology website so that you can get a range of other resources designed to help you in your study of the A-Level Biology course.